Good afternoon. My name is George Adigbli. I'm going to give a talk about retrograde transport and the role of tRNAs in it in the nuclear import of human immunodeficiency virus. My work's from University College London. There are no disclosures. I'm going to start the talk by talking about HIV. I won't go through the specific virology of the virus itself, but I'm going to focus on a few of the important things that I'll be looking at in the structure of the virus. I'll be focusing mainly on the GAG and the GAG pole regions here, which you can see are the core and the matrix proteins. And they're also, the other critically important issue is the reverse transcriptase. So I'm going to look at the retrovirus life cycle. Basically, it starts with fusion of the virus with the membrane of the target cell. You then get viral attachment surface proteins which bind to specific cell surface receptors, leading to the delivery of the virus into the cytoplasm. The completeness of the dissociation of this capsid from the reverse transcriptase complex and the time it takes place are quite significant, and they impact on the later events in the retroviral life cycle. And this is potentially a distinguishing feature which distinguishes between the ability of different retroviral families to infect cells. The capsid protein probably rearranges and or uncoats to expose the reverse transcriptase complex. And after uncoating, the viral RNA is converted to double-stranded tRNA by reverse transcriptase, and at that point it's known as the pre-integration complex. And then after this, you have virion components. We'll move on to the next slide. The pre-integration complex then enters the nucleus, accesses the host chromatin, and the viral DNA integrates into the genome, in which case it's known as the provirus. Proviral DNA is transcribed by the host cell using RNA polymerase II, and this turns into full-length RNA transcript. Viral mRNA is then translated into proteins, and these then bud off and form virions, which bud off from the surface and then begin maturation. So in our study, we're looking at nuclear import and how do retroviruses actually access the host replication material. We're aware of the general mechanisms utilized by retroviruses to traffic through the cell to the nucleus, and of some of the events that occur following transcription. However, the question relating to how it actually translocates into the nucleus is largely unanswered and is the subject of some controversy. Different classes of retroviruses have different characteristics in this regard, and one of particular interest is the varying ability of dividing cells versus non-dividing cells, and the access to of nuclear import. As HIV DNA is able to access the nucleus before mitosis, it's been suggested that its pre-integration complex actually contains proteins with nuclear localization signals, which are essential for its ability to affect non-dividing cells. And these include things such as the matrix protein and the integrase protein. Importin 7's also been seen and documented by Fossati et al. And they think that's a import receptor responsible for movement of ribosomal proteins. I'll look a little bit more into tRNA, which is quite a new concept seen in the nuclear import of reverse transcriptase complexes. We'll start by looking at the dynamics of tRNA. In eukaryotes, tRNA links transcription, which occurs in the nucleus, and translation, which occurs in the cytoplasm. It used to be thought that it was a typically unidirectional phenomenon occurring from the nucleus into the cytoplasm. However, the subcellular translocation is not constitutionally unidirectional. It's more cyclical, which you can see when you look to the right-hand side of the image. And this question gives us a question of whether there's a potential role for tRNA in HIV cellular trafficking. So Said-Siva et al., who worked in our lab at the University College of London, showed that retrograde transport of specific tRNAs promotes nuclear import of HIV. tRNAs can access the nucleus also without transit together with reverse transcriptase complexes. And so we wanted to identify differences between retroviruses that can and can't infect non-dividing cells. So the specific aims of our project, we wanted to make a comparative analysis of the nuclear import capabilities of gamma retroviruses and lentiviruses, which we know have a difference in ability to affect dividing and non-dividing cells. Then we could explore the role of tRNAs in producing the contrasting phenotypes. The discrete objectives were to create 2 GAG chimera plasmids incorporating HIV and murine leukemia virus within a HIV vector backbone and produce corresponding chimeras. Then we wanted to determine the ability of these chimeras, as well as HIV viruses, which we use as a positive control, to infect both cycling and cell cycle arrested cells. We then would extract the RNA from the 3 viruses, analyze the tRNA they incorporate, and identify any differences between them. And finally, this later step, which we tried to complete actually in the last couple of weeks, was to characterize the influence of these different tRNAs on nuclear import of retroviral reverse transcriptase complexes. So our materials and methods. First of all, we wanted to construct the chimeric proviruses. So we took genes from HIV and murine leukemia virus and used proviruses PLA and PAMS respectively. The chimeras were made by precisely replacing HIV sequences with MLV sequences. The sequences at the junctions between MLV and HIV in the chimeras are exactly the same as those in the parental viruses, making the ligation step quite easy. The 3 that we looked at were we had the HIV vector backbone and inserts containing the matrix and capsid proteins of MLV1, complete HIV provirus except for the matrix from MLV in the second, and in the third we had HIV provirus except the matrix protein and the P12 are replaced from MLV. That one we didn't actually go too much into because it wasn't very stable. We then amplified the primers and provided cloning. The restriction digests, for your information, were performed using APA1 and NOT1, which you can see the recognition sites are complementary. The P8.9 N-SB vector fragment is the HIV backbone, and then we ligated the insert fragments. So production of the replication incompetent HIV MLV vectors. We transfected 293 T cells using the DNA that we had created and purified the virus using spins in a 25 to 45% sucrose cushion, extracting the virus from the interface. We then analyzed the RNA after extracting it and ran it on a long 50 centimeter gel using polyacrylamide gel electrophoresis, stained this with cyber gold solution, and then excised the tRNA containing bands and recovered the RNA by ethanol precipitation in the presence of glycogen. The nuclear import assay we use is we use cells 50% confluence, and we permeabilize these using digitonin and wash them twice and incubate for 15 minutes in the presence of labeled reverse transcriptase complexes, our own tRNAs, and an energy regenerating system, because this is an active transport mechanism. We then analyze this by confocal microscopy. The results. First of all, just talk quickly about the production of the chimeras. GAG gene expression determines the ability to infect non-dividing cells. And this has been demonstrated in the literature. So our question was, does GAG participate in specific tRNA incorporation into retroviral particles, and hence does it influence reverse transcriptase nuclear import, reverse transcriptase transcription complexes nuclear import? GAG inserts containing the matrix protein and the P12 and capsid genes cleaved from infectious provirus, cloned into the backbone, and we did diagnostic digest using BSE2. And the reason for this was because we get this was to ensure that our inserts had actually been inserted into the plasmid rather than religation of the native wild-type HIV. Plasmids containing our inserts possess 2 restriction sites for PSTE, whereas the native wild-type virus only had 1. So successful cloning was shown by having 2 bands, as opposed to the 1 frayed band, which would be present if the native HIV had religated. And these are just the structure of the restriction digests. We then wanted to characterize the infectivity phenotypes of HIV and MLV and establish whether the plasmids generated were actually capable of producing infective retrovirus. This may help to explain the phenotypic discrepancies between the 2 subfamilies. We use viruses we use to infect normally dividing HeLa cells, as well as cells arrested in the G1 S phase by fidicolin. Fidicolin inhibits DNA polymerase alpha and arrests cells in the S phase, or prevents them entering the S phase, depending on the cell cycle, the stage of the cell cycle at the time of treatment. And arrest premitosis enabled quantification of the ability to infect cells which had intact nuclear envelopes. So here we look at the first, after 48 hours of incubation with the virus, we analyze the cells by flow cytometry. You can see along the top line A, they have dividing cells, and along line B, these are the cell cycle arrested cells. You have the native wild-type HIV. On the far right, you have the wild-type MLV, and in between you have the matrix chimera and the capsid chimera. And what we've basically shown is that there's confirmation of successful production of the viral vectors capable of infecting cells, and verifies that our HIV, you can see here, is capable of infecting both dividing and non-dividing cells, non-dividing cells with greater efficiency. The graph on the left illustrates the percentage of infected cells at each titration of HIV. The second graph, percentage of infected cells relative to the ability of HIV to infect arrested cells. A similar trend we noticed to HIV was seen in the matrix chimera. However, the infectious titers were less than a fifth of that of native HIV, and the capsid chimera exhibited poor infectious titers in cycling cells and an almost complete inability to infect non-cycling cells, even when we use concentrations 10 times above the rest of them. As you can see, MLV shows complete inability also to affect non-dividing cells. So from this, we can see that MLV, the matrix protein from MLV, can somehow defect the infectivity of HIV. The magnitude is relatively small, and this goes hand in hand with work by Fouchier, who demonstrated using mutant knockout of the matrix that it doesn't aggregate post-entry nuclear import, and so he dismissed a determinant role of MA in nuclear import. The P12 capsid chimera showed P12 and capsid proteins demonstrated dominant negative effect for nuclear import of reverse transcription complexes. We then wanted to examine the tRNA composition of wild-type HIV and chimeric viruses, vectors to correlate this with their infective phenotypes. So to do this, we first needed to prepare highly concentrated purified viral stocks, and then we infected HeLa cells with serial dilutions of each. And here is the results that we get, which suggests that our viruses were suitably concentrated for further use. And it's just we compared it to the previous flow plot you saw before. And also it showed that we maintained our infectivity phenotype, because as you can see, compared to the old one, there's no difference in the general configuration of the bars. So the next question is, are there any differences in the tRNAs incorporated by these different viruses which account for their different infectivity phenotypes? To do this, we extracted RNA from viruses and resolved them on the long page gel, stained it with cybergold. The reason that we used this was to destroy tertiary and secondary RNA structures, which would have affected both migration and our visualization of the bands. This promoted clean separation of our RNA on the gel and facilitated our identification of the tRNA bars. And our results showed that there were quantitative and qualitative differences between the viruses and the chimeras. So this is an image of the gel. And you can see, I'll talk you through. Here we have just a normal type of cell. We have HIV and the two chimeras. And then we have, so this is wild-type HIV and here's wild-type MLV. This is the virus-specific band, and these are the tRNA segments you can see. There was minimal disparity between the tRNA incorporated by HIV and the matrix chimera, and the banding pattern was consistent, as you can see. When looking at the capsid protein, however, the banding pattern was different. It incorporates approximately 17 times less tRNA than HIV. I'll go back to it. You can see that the bars are much thinner. However, when you look at the virus-specific band, you can see that the same concentration was loaded, so actually it is a much reduced amount of tRNA. There's, so as we see, there's minimal disparity between the HIV and the matrix component. This is just a graph to show how we quantified the results using a Storm scanner and QuantiQuest. And what the suggestions were from this is that the amount of tRNA incorporated via the viruses might impact on their ability to infect non-dividing cells. Different amounts or relative compositions of specific tRNAs incorporated may be significant in this regard. And considering the relative infectivity phenotypes that we saw before, we question whether a link between tRNA composition and differences in these abilities to viruses to infect cells. Last thing we looked at was the MLV banding pattern, which was completely different to those from HIV, much darker bands, and the positions of the bands were also distinct from the other viruses. As you can see here, there are multiple bands, you can see 4 here, and there's quite poor demarcation between them. So does tRNA participate in determining the infectivity phenotype? The next question. Indeed, when we wondered whether or not there's a steric hindrance component to this, because there may be incomplete coating of the capsid, which blocks access of the reverse transcriptase complexes to the tRNA. And if this is because of an uncoating property, we wanted to try and see if further work could give us an actual picture. So then we wanted to find out if this putative link between the infectivity profiles and the tRNAs, whether we could see that actually when we're looking at nuclear import of HIV. So to do that, we eluted tRNAs from the gel, and we use these in the nuclear import assay. Fortunately, what we see here, I tried to do this in the last week before this for conference. We got inconclusive results basically. At the bottom along E, you can't really see very well, that we have that's a negative control, and the rest of them are the HIV and the chimeras. And we think that the most likely problem with this, considering that this has been done before in studies Weizsäcker, was that we're basically seeing that there's only background distribution of the RTCs because there's no difference with the negative control. It's most likely due to insufficient quantities of tRNAs used in the import assays. Fortunately, that was all that we could use. We loaded 300 nanograms onto the gel, whereas other studies in the nuclear import assay actually use 100 nanograms. And if you look back at the gel itself, you can see that what we were getting in this portion is quite a lot less than 1/3 of the thickness of what was run in the gel. So we probably lost quite a lot there. The other thing is that it could be a problem with our reverse transcriptase complexes, because we noticed that even the positive control didn't show any difference. Oops, sorry. Yeah. So our reverse transcriptase complexes may have degraded, so we'll need to repeat it again on that point. So in discussion, we noticed that there is a negative correlation, and this is as suggested in the literature. There was an article by Yamashita and Emmerman who demonstrated the incorporation of MLV capsid into a HIV backbone markedly abrogated the ability of viruses to infect non-dividing cells. Capsid does seem to be a dominant determinant of retrovirus infectivity in dividing cells. Differences in the tRNAs incorporated primarily are quantitative, and we saw this most when we looked at the capsid. The genotypic differences accompany a reduced ability of this capsid virus to infect non-dividing cells. So we wonder, is the effect of tRNA specifically related to the reverse transcriptase complexes? And if so, that's weight's given to that by the fact that we think that there is a steric hindrance effect. Further work would be to repeat the experiment looking at nuclear import again more specifically, and also to characterize the specifics of tRNA and the interaction with the viral proteins and the RTC. These are the references that we used. Thank you.